Thanks, everybody. Thank you, Jeff and the Invivoscribe team for inviting me to be here today. It's a great pleasure to be here and to share with everybody some of the research work that we've been doing using LymphoTrack in plasma DNA. So I don't have a disclosure slide. I don't have any relevant disclosures. So this is the outline to the talk. First, I will introduce sort of the concept of plasma DNA, the sort of composition of it, and why it's really important to understand the molecules before we can even think about applying it to B-cell lymphoma. Next, I'll talk about sort of the use case that we're studying this in terms of high-risk populations, and then finish with the pre-analytical considerations that I think anybody trying to move into this area should think about, and then finally some of our preliminary data. So plasma contains sort of a variety of circulating proteins, nucleic acids, and even exosomes. It's sort of the process of normal cell turnover will release all of these molecules into circulation. And like I said, normal, but also when there is an abnormal state like cancer, those cells also will undergo turnover and release these molecules. And in those situations, we may be able to identify cancer, quote unquote, tumor-specific abnormalities like small mutations, rearrangements, and the like. However, if you're trying to study cancer DNA, ctDNA, you have to understand they're very rare. They account for approximately only 1% of all the free DNA in circulation. So there's an actual definitional difference between what we call cell-free DNA, CF, and ctDNA. And you can see on the right there, the quantity in circulation really vary a lot. Most people have very little, and the per ml is per ml of plasma, so of plasma volume. Whereas in cancer patients, they can actually get pretty high. Now, this amount actually tends to correlate with disease stage, and even tumor metabolic volume. Most of these studies to date have been done in solid organ malignancies because sort of the difficulty in studying solid tumor in liquid phase, right? So unlike the hematopathologists in the room, you know, I know myself, leukemia is a very easy disease to study because they circulate. And so for our solid tumor colleagues, they've been they grasp onto the ctDNA idea because of the ability to study, not in a non-invasive manner, the genetic abnormalities. And these are sort of the two main areas of application that have been studied both in the research realm in clinical trials and have now moved into clinical practice in terms of how to use plasma DNA. So detection, certainly, right? Identification, prognostication, targeted therapy detection as well, but also monitoring, right? So monitoring disease response, non-response, relapse, the like. And I put in gray the two sort of categories at the bottom, the detection of early stage disease and the screening of high-risk populations, because while these are very active areas of study in the research side, they remain sort of controversial in terms of when and where we can apply this, and I will get into that. And funny enough, it's the exact topic of my talk today. So what I will talk about, which is less controversial, is the use of ctDNA in terms of monitoring. So in B-cell lymphoma, which is our area of study, the amount of, or the type of circulating tumor molecules have correlated with disease state. So you can see here, this is a study out of Stanford where they did both mutational analyses, which is the line in red, and immunoglobulin, which is the line in blue. And so both of these tumor markers, if you will, track with disease, both with response and also relapse. So these are time points where the ctDNA findings predated the imaging, PET-MR, evidence of relapse disease. But how can we use these tumor markers in assessment of people who are known to be healthy but who may have cancer? And so this hospital, anybody know, well, I put where it is, but they probably don't know the hospital name. This is Chris Hani Baragwanath Hospital in Soweto, South Africa. It's actually the largest hospital in Africa, in the entire continent. It's about a 3,400 bed hospital located on 170 acres. So a huge plot of land. And you can see these sort of barrack style buildings. These are actually where the patient floors are. So it's not tall buildings like we think about it, but it's all flat. And the line there, I drew the line because that's outside of the line is the town of Soweto, South Africa. And then inside that line is actually the perimeter of the hospital. And so I bring this up because this is actually the population that we're studying. We have active collaborations with investigators there. So the hospital is one of the main teaching hospitals of the University of Witwatersrand in Johannesburg. And in Johannesburg, the average HIV infection rate is 20% of individuals. And actually, their lymphomas are more diagnosed in HIV positive people than in HIV negative. And so our group is really interested in studying in HIV patients or people living with HIV, sort of, which is a much higher risk. They have a much higher risk for lymphoma, whether these plasma markers can actually assist with the initial diagnosis. And the reason we think plasma markers may be helpful is that there's a particular aspect of the diagnostic sort of odyssey there that is lacking in this country, which is that the diagnostic interval can be exceedingly long. For example, our colleagues there have noted that the typical diagnostic workup of HIV people with sort of B symptoms and lymphadenopathy is that they will go to an FNA clinic. And the main purpose of the FNA clinic is actually to test for TB, because TB is the #1 cause of death in HIV individuals. It's a very high public health and morbidity mortality concern. And so what happens with the aspirate is it goes for actually a molecular analysis for TB, and the patients are typically put on empiric TB treatment in the meantime. Now, FNA, the aspirate alone, may actually aspirate out cells that are malignant appearing or actual cancer cells. However, that knowledge is typically not known for about another 2 months or so. And so that's already a 2-month period, a gap, and oftentimes these patients are on TB treatment for a prolonged period until symptoms don't regress or that persistence and worsening of symptoms bring them back to medical attention. And eventually a surgical biopsy is performed. However, the infrastructure is so overburdened that the average time to biopsy, not even to a diagnosis, is usually 12+ weeks. And so our goal with our study is to bypass potentially this entire middle ground here and say, at the time of presentation to these FNA clinics, could we have a plasma marker test that could triage appropriately these patients so that they are not waiting 2 months to fail TB therapy? And we believe that the prevalence of lymphoma in this population is up to 5%, so 5 individuals out of 100. And these FNA clinics see 200 HIV positive people with lymphadenopathy each month. So you potentially are identifying 10 HIV-related lymphomas. So we think the motivation is strong and sort of the impact could be very large. However, we have a problem. We do not have a tumor in hand. We do not know what the tumor characteristics are, genomic characteristics are. And so how do you, de novo, sort of decide what's an abnormality and what is not? And so the rare paucity, right, of the ctDNA really requires an enrichment for that signal versus noise, what's signal versus what's noise. And so we have a few options. So in studying HIV individuals with lymphoma, we often can find virally driven lymphoma, such as EBV or HHV, and we have sort of exploited that in terms of as a tumor marker. However, there are other sort of somatic or host genome, right, abnormalities that can be found in the lymphoma and as such. So today, before we get to B and T cell gene ranges, I want to highlight something in terms of small mutations because this sort of brings home the point of how rare these events actually are. So these are data pulled from several large studies, both hematologic and non-hematologic, which shows you that the amount of ctDNA, if you define ctDNA by the tumor VAF, right, variant allele frequency, the amount of tumor DNA really varies by tumor type and stage. So on the left, we have classical Hodgkin lymphoma, about 3% on average. And diffuse large B-cell lymphoma is actually quite abundant, up to about 10% of the circulating cell-free molecules are tumor-derived. However, when we move into solid tumor cancers like pancreatic and earlier stage solid tumor malignancies, those frequencies drop dramatically down to the 0.1 to 0.0001%. And so really, if our colleagues that are studying solid organ malignancies, they really need ultrasensitive techniques. Whereas for me, I find that very sort of lucky that when we're studying heme malignancies, sort of a 1% maybe LOD could be sufficient, at least a diagnosis. And so knowing how rare it is, we are thinking, how can we increase signal, right? Maybe if you decrease your denominator. So this is why we moved to B-cell clonality. If instead of assessing all of the cells, or all of the DNA, I keep referring to them as cells, all of the DNA in circulation, if you only assess the B-cell DNA, is it easier to find a clone? The answer is maybe. So what we call this is tumor uninformed, right, clonality analysis. So tumor-informed clonality analysis 100% of the time outperforms uninformed analysis. And if you look at studies that have been performed on archived plasma, so frozen plasma, not necessarily collected for cfDNA analysis, you see that our group's work in HIV lymphoma and others' work in non-HIV lymphoma really mirror each other. When you know the sequence of the molecule you're looking for, almost 100% of the time you find it in circulation. However, when you don't know it and you're trying to do de novo calling, it's actually very hard. So maybe 50, if you're lucky, 50% of the time. So really, this uninformed analysis requires abundant ctDNA. So how can you get more ctDNA? So this is what we have embarked upon, is to, again, limit the background cells by using LymphoTrack. So if you look at what has been used on archived plasma specimens in past studies, these are on the left. We have used this PCR, so the traditional PCR method. This is another NGS method that has been developed, which is a very similar framework 1, 2, 3 type of configuration. If you look at these two methodologies that have been used in the past, the LymphoTrack assay actually is designed very similarly. So framework 1 family primers, 2 and 3, and those are the rough amplicon sizes. And so those amplicon sizes become important because do you actually have fragments in circulation of DNA that's of the appropriate size? So before we get to that, we have to think about pre-analytical parameters. So like I was saying earlier, unless a specimen was collected specifically for cfDNA, what you find in most biorepositories, what's called plasma, is a standard collection. So frozen plasma typically is siphoned off from a EDTA tube that was primarily collected for cells, right, for genomic DNA. And those, the delay could be up to 4 days or even 7, 8 days. It's really uncertain. And the spinning, so the initial processing is one spin, the cells are separated, the plasma is taken off, frozen, you're done, right? So actually that is actually not the preferred method of studying cell-free DNA, and I'll show you why. In cell-free DNA studies, most of the EDTA tube studies have had really rapid processing times, within 2 or 3 hours. I mean, you can stretch it to a max with 6, and you can maybe even stretch it to 24 hours if you put it at 4 degrees. However, ideally it's within 6 hours. And the spinning this time is now 2 spins. So initial spin to pellet, and then a secondary spin to pellet any additional particulates. What has really revolutionized, I think, the study of cfDNA is the Streck tube, which is a little bit of fixative, and we don't really know what kind, but is able to allow storage at room temperature, so ambient room temperature, for 14 days, up to 14 days. And so we really moved towards the Streck tube, given our sort of setting. Transportation is not really consistent where we are, and so we moved to the Streck tube when we realized there were these limitations. And so now that we think we have the right tube, we want to know what are our benchmarks, right? What does high quality cfDNA or ctDNA really look like? So this is the structure of a nucleosome core. And the idea is cells that die by apoptosis will fragment in a predictable manner. In that, the mononucleosome, which is 1 turn, plus the linker, should be the majority of the size of the cell-free DNA in circulation. Whereas sometimes, occasionally, you get these dinucleosomal molecules, which are 2 times, right? So those are the expected sizes. However, in reality, what we see is that most cancer patients, their cell-free DNA actually is frequently less than 150 base pairs. That mononucleosome fragment is actually much smaller than predicted. And you can see here healthy, low ctDNA cancer and high ctDNA cancers. The higher the ctDNA amount, the more fragments under 150 base pairs. So that's probably what we're shooting for. And what do we see? Well, we realized that optimal processing really made a big difference. So if you look at the concentration, just the straight maybe qubit reading, the concentrations may look very good in a frozen plasma, for example, on the right, that was extracted. However, when you run it on a high sensitivity gel, you're able to see the actual profile and distribution of the molecules. And then you realize that, for example, you can't really see the scale here, but on the right-hand side, the scale goes up to 10 fluorescence units, whereas on the left it says several thousand. So you get a lot more DNA when you do the proper upfront processing. But the other thing you notice is now you can actually see the mononucleosomal and the dinucleosomal peaks, whereas you can see it over here, but they're much more attenuated. However, no matter what magic this Streck tube has, it cannot rescue compromised specimens. So the typical practices of clinical lab medicine, where you don't accept hemolyzed, lipemic specimens, still holds true here. So you can see on the left, that was a hemolyzed specimen where we got almost all genomic DNA and degraded DNA, right? And no cfDNA. On the right, similar story, lipemia. But when we did this processing optimization and numerous rounds of extraction optimization, we finally found that when we compare the archived specimens, which are in red, and the prospectively collected specimens in blue, we found that majority of the DNA we were recovering in the optimization tubes was probably cell-free DNA, 85%. I'm not saying they're tumor DNA, but they're cell-free DNA. And the amount ranged from 20, 30 nanograms per ml of plasma up to this case, several cases of almost 150 nanograms per ml of plasma. And the dominant fragment length, like I was saying before, most of the dominant fragment lengths in this situation were around 130 base pairs, so less than 150, what we wanted, whereas the archived plasma had many more molecules greater than 150. And this may have been the result of sort of multiple freeze thaws of the specimen over time and actual degradation of genomic DNA. So now that we had high quality DNA, we were actually able to perform all three frameworks of the IGH LymphoTrack analysis. So I'm showing here total reads in the framework 1 analysis, framework 2, and framework 3, and we have NTC, the negative and positive controls, and then the plasma. So on the left-hand side, you can see that this line here is drawn at 10,000 reads, so we do everything in replicate, so in total we have 20,000 reads. But at 10,000 reads, the framework 1 reactions are actually almost approaching 10,000 reads for something that's about 300 or so base pairs up to, right? Then in the middle, we see that almost all of the framework 2s exceed that minimum. And then on the right, framework 3, which is sort of maybe a little bit confusing when you look at this. First off, NTC, why is there so many reads? Actually, none of those are mapped. So if you look at the actual mapped reads, they're not there. Secondly, you can see that as compared to the archive frozen plasma, the total reads of the optimized plasma may actually be a little bit lower, right? So it's counterintuitive because it may actually be a little bit lower because it's more specific. So what we saw was, for example, if you look at our clonalities display, and so we actually do it in a combined molecular weight-based and sequence-based method. So we have molecular weight and base pairs on the x-axis. Each color is a single molecule or sequence. And then on the y-axis, we have percent read representation. And you can see that after optimization, the scale here is 80%. The scale here is maybe 4%. So you have much less noise, right? And so the more reads you got may not have been cell-free DNA, may actually have been degraded genomic DNA, i.e. B cells in circulation. So we think now that you have a clean background, the clonal dominance becomes much more apparent. So we actually don't have a cutoff that we figured out. So we're working on this actively, and we're trying to do a classification regression tree analysis at the end of our study when we have collected all the samples to tell us what a real cutoff should be. However, when you look at this, there is no doubt in my mind that this is a clonal sample, and this is a, yes, clonal sample, but with a polyclonal background. And so the clonal dominance becomes much higher and much easier to tell once you have very high quality DNA. And so these are our final results. Of the 9 specimens we have run so far, uninformed analysis gave us an overall sensitivity of approximately 56%. And you can see, if we were limited to framework 3 alone, we would only identify 3 clonal samples in 9 lymphoma patients. And so we are working on improving the sensitivity, obviously, for the 4 samples that we don't have, and for future samples, and we're thinking about using IGK or leader analysis. And now because we have excess DNA left over from many of these samples, and high fragment size DNA, we're probably able, we haven't seen it, but we're hoping we're able to do these analyses. And so going back to the question of, can plasma cfDNA assessment improve diagnostic yield? And we believe that given some of the limitations, including somatic hypermutation that you see in B and T cells, that the receptor analysis is probably not enough. And so we're approaching this in a more multi-analyte approach with looking at viral sequences, methylation, mutation, and also CNV analysis. So this is actually a CNV plot generated from NGS data that we performed on one of these 9 patients. This is a patient with Burkitt lymphoma. And you can see that the copy number analyses actually show very clearly there are distinct copy gains and a homozygous copy loss of cyclin cyclin CCKN2A. And while if I did not know that this patient had Burkitt lymphoma, even if I didn't know that, I would look at this and be highly, highly suspicious that this patient should be triaged for immediate excisional biopsy. And so we have not so far been screening, doing this in a screening setting. This has been a pilot study, and so this is sort of the overall goal of our study, is to be able to compare cases and controls in a multi-analyte approach, both EBV, methylation, and mutations, to create a diagnostic algorithm. And on the right, we have sort of outlined imagine, this is not real data, an imagine card analysis tree where you could walk yourself down based on the result at each stage and get to either a very high confidence that this could be lymphoma or a very low confidence that this could be, in this case, an EBV lymphoma, and potentially be able to appropriately triage patients that way. And so the summary here is, for those of you wanting to embark into this space, plasma DNA is a very valuable source, I think, of tumor DNA. However, they are rare, so you must think about sensitive and specific assessment techniques, and that the upfront processing is sometimes more important than the downstream analysis, and that only with the improved quality and quantity of DNA can you really do a lot of the studies you're aiming to do. However, our experience has been that the LymphoTrack technique has actually been very helpful in this regard. And so these are the folks I'd like to acknowledge, including people in the audience. Thank you so much. And this work was funded by the CFAR grant at Johns Hopkins. And thank you, and questions.